The cell cycle is the process of when a cell grows and divides. During the cell cycle, a cell grows, prepares for division, and divides to form two daughter cells. The cell goes through three processes called interphase mitosis and cytokinesis to divide into two daughter cells. Interphase is the growth period of cells between cell division. The first stage of interphase is called the G1 phase or the GAP1 phase. During the G1 phase, cells do most of their growth in size. It grows to almost its full size. Also, new proteins and organelles are synthesized. Individual chromosomes are not visible in this phase because chromatin in the nucleus is spread out. The next stage of interphase is called the S phase, also known as the synthesis phase. During the S phase, chromosomes are replicated and new DNA is synthesized. At the end of this stage, each chromosome has two molecules of DNA. The final stage of interphase is called the G2 phase or the GAP2 phase. G2 is normally the shortest phase. During the G2 phase, the organelles and molecules required for cell division are made. Also, the cell grows in size. Once the G2 phase is complete, the cell is ready for division. The next process in the cell cycle is mitosis. The first phase of mitosis is called prophase. Prophase is usually the longest process of mitosis. During prophase, the genetic material in the nucleus condenses and we are able to see the chromosomes. The centromere is where the chromatids are attached. A chromatid is one DNA strand in a duplicated chromosome. Also, a spindle, which are a collection of microtubules, start to build out of a region called the centrosome. These spindle fibers help separate the duplicated chromosomes. Towards the end of prophase, the chromosomes are tighter and the nuclear envelope breaks down. The second phase of mitosis is metaphase. It is normally the shortest process. Metaphase is the aligning of chromosomes across the center of the cell. The centrosomes are at opposite poles of the cell. The spindle fibers connect the centromere of the chromosome to each centrosome at each pole. The next phase of mitosis is anaphase. During anaphase, the chromatids split to become individual chromosomes. One chromatid from each chromosome goes to opposite poles in the cell. The spindle fibers are attached to the centromere and help pull and push chromatids. At the end of anaphase, there are two separate groups of chromosomes at each pole of the cell. The last phase of mitosis is telophase. During telophase, the spindle breaks and the chromosomes begin to uncoil and turn into chromatin. A nuclear envelope forms around each set of chromosomes and the nucleolus starts to become visible in each daughter nucleus. The last process of cell division is cytokinesis. It normally occurs at the same time as telophase. During cytokinesis, the one cell splits into two. In animal cells, the cell membrane draws inward and splits into two. In plant cells, a cell plate is formed between the nuclei. The cell plate gradually becomes into cell membranes. A cell wall forms and separates the two membranes. Finally, each new cell created goes through interphase, mitosis, and cytokinesis all over again. Cancer is caused when cells divide uncontrollably. The replicated cancerous cells move to different parts of the body to create tumors. That eventually results in cancer. The cell cycle is much faster when it has cancerous cells. Normally, the interphase period in cancerous cells is much faster than normal cells. Cancer is caused when proto-oncogenes become oncogenes through mutations. Proto-oncogenes control the division of cells. On the other hand, oncogenes stimulate cellular growth. Also, cancer is caused when tumor suppressor genes stop working. Tumor suppressor genes also regulate cellular division.